Hello, friend. Welcome to Science for Sleep. Whether you have joined us many times before or are discovering this quiet corner for the first time, this is a place to slow down, learn something gentle, and allow the day to slowly loosen its hold. Tonight, we are spending time with lilacs. This subject was suggested by one of our listeners, Audrey, who asked for a calm journey into the science of these familiar spring flowers. A lilac bush can seem almost empty for much of the year. Its grey-brown branches stand through winter with no leaves, no fragrance, and no sign of the purple clouds that will appear later. Then, after enough cold has passed and enough warmth has gathered, the change can happen quickly. Small buds swell along the stems. Green leaves begin to open. Tightly folded flower clusters push outward. Within days, a shrub that looked quiet and bare can hold hundreds, or even thousands, of small flowers. The common lilac is known to botanists as Syringa vulgaris. It is a deciduous shrub, which means it drops its leaves each year. It belongs to the olive family, the same large plant family that includes olive trees, ash trees, jasmine, privet, and forsythia. Wild common lilacs come from southeastern Europe, especially the northern Balkan Peninsula and parts of Romania. In gardens, mature shrubs often grow between 8 and 16 feet tall, with many woody stems rising from the same base. If you enjoy these quiet journeys, you are welcome to subscribe. And if it feels natural, to leave a comment sharing where you are listening from tonight. It helps others find this calm corner of science, one sleepy listener at a time. The flowers may be the part we notice, but they are only the final visible stage of a process that has been unfolding for many months. A lilac does not begin building its spring flowers when the weather turns warm. The basic flower structures are prepared inside buds during the previous growing season, often beginning in summer. By autumn, those future flowers are folded into small packages along the branches. Protective scales close around them. The leaves fall. The shrub enters winter carrying much of the next spring already inside it. Those buds cannot safely open during every mild spell. A few warm days in November may be followed by deep cold, ice or snow. If tender flower tissue expanded too early, it could be damaged before winter had truly begun. Lilacs avoid that danger through dormancy, a controlled state in which growth remains paused, even when the weather briefly becomes mild. Long exposure to cold gradually changes the condition of the buds. Once enough winter chilling has occurred, the buds become more able to respond to spring warmth. The shrub is not reading a calendar. It is responding to temperature, water, light, and chemical signals inside its tissues. As warmer days accumulate, cells inside the buds begin taking up more water. Their internal pressure rises. Small branches inside the flower cluster lengthen. Petal tubes stretch. The compact structure slowly unfolds. This is why lilacs are closely tied to places with cold winters and clear seasonal change. In regions where winters stay too warm, many common lilacs grow poorly or produce fewer flowers because their normal cold requirement is not fully met. In a suitable climate, however, months of preparation lead to a bloom that may last only a few weeks. That brief display has a large purpose. The flowers produce color, scent, nectar, and pollen during a narrow period when visiting insects can move between them. Afterward, the petals fade. Leaves take over the work of gathering energy, and the shrub begins preparing for another year. What looks like a sudden burst of purple is really the meeting point of two seasons. It carries work completed during the previous summer, protection maintained through winter, and growth released by spring. And even the large purple cluster is not one flower. It is a branching structure made from many tiny flowers, each with its own tube, petals, pollen, and reproductive parts, all gathered closely enough to appear as one soft cloud from a distance. At close range, the soft purple mass begins to separate into smaller shapes. Each tiny bloom has its own petals, tube, pollen, and central reproductive parts. Together, they form an inflorescence, which is the botanical name for an organized group of flowers carried on one branching system. In the common lilac, this structure is usually shaped like a narrow cone or pyramid. Botanists call it a panicle. A main central stalk runs through the cluster. Smaller branches extend from that central line. Each branch divides again and carries several individual flowers, often called florets because they are small flowers within a larger display. A typical common lilac panicle may measure about 6 to 8 inches from base to tip. Some remain narrow and upright. Others spread more widely or bend slightly beneath the weight of their flowers. The exact form depends on the species, cultivar, weather, and age of the branch that carries it. The individual flower is much smaller than the cluster suggests. Most common lilac flowers are less than 1 inch across. Their petals are joined near the base, creating a narrow tube called the corolla tube. The word corolla refers to all the petals of a flower considered together. At the upper end of the tube, the joined petals separate into 4 spreading lobes. These 4 rounded sections create the familiar cross-like shape seen in a single lilac flower. Beneath the corolla sits a much smaller structure called the calyx. The calyx is made from leaf-like parts called sepals, which protect the young flower while it is still developing. In a lilac, the calyx forms a small cup with 4 shallow teeth around its edge. Inside the corolla tube are 2 stamens. A stamen is the pollen-producing part of a flower. Each one ends in an anther, where the fine pollen grains develop. The stamens are described as inserted because they are attached to the inner wall of the corolla tube rather than standing separately from it. The pistil rises through the middle of the flower. This central structure includes the stigma, which receives pollen, the narrow style beneath it, and the ovary at the base. Ovules are held inside the ovary. After successful pollination and fertilization, some of those ovules can develop into seeds. The 4-lobed form is typical of wild common lilacs and many traditional garden varieties. Breeders have also preserved double-flowered cultivars. In these forms, additional floral organs have developed into petal-like structures. Each floret looks rounder and fuller, sometimes appearing to contain a smaller flower nested inside the first one. A double lilac may seem especially dense because both the individual florets and the full panicle carry extra visual weight. The flowers within one panicle do not always open at exactly the same moment. Florets near one part of the cluster may already be fully spread, while others remain as narrow-colored buds. This staggered opening helps the display remain active across several days. Fresh flowers continue producing scent and offering nectar, while older ones begin to fade. One panicle, therefore, contains several stages of flower development at once. Certain cultivars greatly expand this branching arrangement. Lilac Sunday, developed at the Arnold Arboretum in Massachusetts, produces flower clusters near the ends of its branches and at many points farther down the stems. A flowering branch can create a display extending more than 2 feet. From a distance, it may resemble one long plume. Closer inspection reveals repeated panicles, smaller branches, and hundreds of separate florets arranged along the woody shoot. This architecture increases the plant's visibility to flying insects. A large patch of purple is easier to locate than one flower less than an inch wide. The many corolla tubes also concentrate nectar and scent within a compact feeding area. An insect can move between nearby flowers without repeatedly leaving the cluster. Each visit creates another chance for pollen to attach to the visitor's body and reach the stigma of another flower. The whole panicle functions through the combined effect of its smaller parts. Its color comes from many separate petals. Its fragrance rises from many small floral surfaces. Its reproductive potential is spread among many individual ovaries and pollen-bearing anthers. Every one of those structures requires water, dissolved minerals, sugars, and firm support. Those supplies arrive through the branches beneath the flowers, connecting the brief spring display to a woody organism that can keep growing for many decades. The narrow stalk beneath each panicle joins a young green shoot, which then connects with thicker branches formed during earlier growing seasons. Those branches carry the weight of the flowers, while also serving as transport routes between the roots and the canopy. A mature common lilac often grows as an upright shrub, with several main stems rising from the same base. Many specimens reach between 12 and 16 feet tall and spread roughly 8 to 12 feet across, although dwarf cultivars remain much smaller, and old plants can exceed those usual dimensions. Common lilacs are woody perennials. Their living framework remains in place from year to year, even though they shed their leaves each autumn. The term deciduous describes this yearly loss of foliage. After the leaves fall, the branches remain alive through winter, holding dormant buds along their surfaces and stored energy within the shrub. New shoots may also rise from the base or from connected underground tissues. These shoots are often called suckers. Over time, suckering can turn one planted lilac into a wide cluster of stems with different ages and thicknesses. A young stem may have smooth green or brown bark. Older stems become grey, rougher, and more heavily branched. Inside them are several layers of specialized tissue that keep the shrub supplied. Water enters mainly through fine roots growing among soil particles. Those roots also absorb dissolved mineral nutrients, including nitrogen, phosphorus, potassium, calcium, and magnesium. The water and minerals enter a transport tissue called xylem. Xylem forms long internal pathways that run from the roots into the stems, branches, leaf stalks, and flower clusters. Much of this movement travels upward as water evaporates from the leaves. That evaporation creates a pulling force through the connected columns of water inside the plant. Root pressure and the attraction between water molecules also help maintain the flow. By the time water reaches a flower near the top of the shrub, it may have traveled several feet through microscopic tubes. The same supply keeps petals firm, carries minerals into growing cells, and replaces water lost to the surrounding air. A second transport system, called phloem, moves many of the sugars produced by the leaves. Phloem lies closer to the outer part of a stem than the main water-carrying xylem. It distributes sugars toward tissues that are actively growing or storing energy. Developing shoots may receive part of that supply. Roots use another portion for growth, maintenance, and storage. Future buds also depend on sugars delivered through this network. The lilac's leaves begin expanding around the flowering period and continue working after the petals have faded. They grow in opposite pairs, with one leaf attached on each side of a stem at the same level. Most are simple leaves, meaning each leaf has one complete blade rather than several separate leaflets. Their shape is often ovate or heart-shaped, with a broad base and a pointed tip. Sunlight entering these leaves powers photosynthesis. During this process, chlorophyll absorbs light energy. The plant uses that energy to combine carbon dioxide from the air with water brought up from the roots. The resulting reactions produce sugars that can be used immediately or converted into other compounds. Oxygen is released as part of the process. Photosynthesis continues through much of the growing season, long after the spring flowers have disappeared. The shrub then directs its resources toward extension growth, root maintenance, wood formation, tissue repair, and storage. Sugars may be converted into starch and held in roots, stems, and other living tissues. Those reserves help support early growth the following spring, before the new leaves have fully expanded and begun producing large amounts of fresh sugar. Lilacs also add new xylem each year through a thin layer of dividing cells called the vascular cambium. This growth increases the width and strength of the stems. Older xylem gradually becomes wood, creating a more durable framework for future branches and flower clusters. Different members of the lilac genus arrange this framework in different ways. The Japanese tree lilac, known as Syringa reticulata, can develop one prominent trunk or several large trunks and may reach the size of a small landscape tree. The common lilac usually retains its broader multi-stem form. Both depend on the same basic partnership among roots, transport tissue, leaves, and stored reserves. Their flowers remain visible for a short part of the year, while the supporting organism continues working through every season. Soon after the current bloom ends, part of that stored and newly produced energy is directed into young tissues, where the first stages of the following spring's flowers begin to form. By early summer, the purple flowers have begun to lose their color. Some panicles remain on the branches as dry brown clusters. Others have been clipped away soon after flowering. The leaves are now fully expanded, and their broad green surfaces continue gathering sunlight through the warmest part of the year. Sugars made during photosynthesis move through the phloem into growing shoots, roots, and storage tissues. Part of that energy is also directed into buds that will not open for many months. Along each new shoot are small growing points called meristems. A meristem is a region of young cells that can keep dividing and produce new plant structures. Some of these growing points continue building leaves and stems. Others begin changing toward a reproductive role. This change is called flower bud differentiation. Differentiation means that unspecialized young cells begin developing into particular structures with particular jobs. Inside a future flower bud, cells gradually organize into the branching framework of a panicle. Smaller flower-bearing branches begin taking shape along a central axis. The earliest parts of individual florets then appear in a fixed order. Protective sepals form around the developing flower. Petal tissue begins organizing inside them. Stamens and the central pistil follow as the bud becomes more complex. None of this can be seen easily from outside. The developing structures remain compressed into a space that may be only a few millimeters long. Studies of common lilac have found that natural flower bud formation often begins around July. Development continues through late summer and into autumn. The timing varies with climate, cultivar, plant health, and local weather, but the yearly pattern remains clear. The flowers seen in spring are prepared well before winter arrives. Lilacs are therefore described as flowering on old wood. Old wood means shoots that grew during an earlier season, rather than soft stems produced immediately before flowering. A branch formed this summer may carry flower buds that open the following spring. This explains why pruning time has such a large effect on bloom. Cutting a lilac shortly after the flowers fade gives the shrub time to produce new shoots and prepare future buds. Pruning in late autumn, winter, or early spring can remove buds that already contain much of the next flower display. The shrub may remain healthy. Leaves can still open. Roots can still grow, and new branches can still form. The missing flowers result from the physical removal of reproductive buds rather than a failure of the whole plant. A heavily winter-pruned lilac may therefore produce strong green growth with very little bloom. Gardeners sometimes mistake this response for poor soil or a lack of fertilizer. The explanation may be visible in the cut branch tips where prepared buds once rested. As summer continues, the buds become firmer and more enclosed. Overlapping bud scales form around the developing tissues. These scales reduce water loss and help protect the soft structures from rubbing, drying winds, and sudden changes in temperature. Their outer surfaces become tougher as autumn approaches. The future flowers remain alive inside, supplied by the branch but held in an increasingly protected state. Shortening days and cooler nights begin changing the shrub's internal chemistry. Growth slows. Leaves gradually stop producing as much sugar. Nutrients are recovered from aging foliage before the leaves separate from the stems. The flower buds continue moving toward a deeper state of dormancy. By late autumn, much of the structure required for spring flowering has already been established. A small bud may contain the beginning of a panicle, the outlines of many florets, and tissues that will later produce color and scent. The shrub has invested water, minerals, and stored carbon in a season it has not yet experienced. Snow depth remains unknown. Extreme cold may arrive. A warm spell could interrupt the normal sequence. Yet the buds must be prepared in advance because building an entire flower cluster after winter would take valuable time during the short spring flowering window. Preparation creates speed, but it also creates vulnerability. The compact flowers must remain closed through every mild autumn afternoon and every temporary thaw. Their survival now depends on a physiological lock that keeps growth paused until a long period of winter cold has changed the condition of the bud itself. As autumn cools, the lilac's leaves turn yellow and separate from their stems. The flower buds remain attached to the bare branches, enclosed by firm overlapping scales. Inside each bud, the future flowers are still alive. Their cells continue carrying out basic chemical reactions, repairing damage, and maintaining the membranes that surround them. Growth has simply been reduced to a tightly controlled minimum. This resting condition is called dormancy. Dormancy protects the lilac from the unstable weather of late autumn. A mild spell may raise the daytime temperature well above freezing for several days. Without an internal restraint, the prepared flower tissues could begin expanding, as though spring had arrived. Their protective scales would separate. Soft tissues filled with water would become exposed. A return to deep cold could then freeze those cells and damage the entire flower cluster. Lilacs reduce this risk through a stage of dormancy in which the buds remain unable to grow normally, even when they are temporarily moved into warm conditions. Botanists often call this internal state endodormancy. The prefix endo means within, because the restriction comes from processes inside the bud itself. Plant hormones help maintain this condition. One important hormone is abscisic acid, which becomes associated with slower growth and stronger dormancy in many woody plants. Changes in gene activity also reduce cell division and alter the way buds respond to warmth. The bud scales provide a physical layer of protection, while this internal control limits development. Their dry outer surfaces reduce water loss and shield the compact flower parts from winter wind. The small size of the bud also helps. A tightly folded structure exposes less tender tissue than an open panicle several inches long. As winter continues, repeated exposure to cool temperatures gradually changes the bud's physiology. This process is often described as the accumulation of chilling. A chilling hour is one way researchers estimate the amount of useful cold a plant has received. Some models count hours within a selected cool temperature range. More complex systems give different values to different temperatures and may subtract value during warm periods. These models help compare climates and predict plant development. But the lilac contains no clock counting individual hours. Its cells respond continuously to temperature through changes in enzymes, hormones, membranes, and gene activity. Cold exposure slowly weakens the internal barriers that have kept the bud dormant. The amount required differs among lilac species and cultivated varieties. A common lilac adapted to a northern climate may need a longer or colder winter than a selection bred for a milder region. Age, plant condition, and the temperatures experienced before full winter can also influence the response. For this reason, there is no single number of chilling hours that applies to every lilac. Nurseries have developed cultivars that flower more reliably in regions with warmer winters. Some are described as low-chill lilacs because they require less cold than traditional common lilac varieties. They still depend on a seasonal temperature pattern. In very warm climates, common lilacs may grow leaves but bloom weakly, open unevenly, or fail to produce a dependable display. Their prepared flower buds have not received the winter conditions to which their development is adapted. After enough chilling has accumulated, the bud enters a different stage. the internal block has largely been released. Growth can now resume when the environment becomes suitable. Botanists often call this eco-dormancy. The prefix eco refers to the surroundings. The bud is capable of opening, yet low outdoor temperatures continue holding it closed. This distinction helps explain why a lilac does not usually bloom during a mild January afternoon. The bud may have completed much of its chilling requirement, but one warm day provides too little sustained heat for full development. Its tissues remain protected while winter conditions continue. Cold hardiness also changes during this yearly passage. A deeply dormant bud can survive temperatures that would injure the same tissues after growth has resumed. Once the scales begin separating and water-rich flower parts start expanding, their resistance to freezing decreases. A late frost in spring can blacken young shoots, damage swelling buds, or destroy flowers that were already close to opening. The winter bud therefore passes through a sequence of changing abilities. Early dormancy prevents premature growth. Accumulated chilling removes the internal restraint. Continued cold then delays expansion until warmer conditions persist. Winter has carried the future flowers through the most dangerous part of their schedule. The buds are now capable of growth, but their exact opening date depends on the warmth that begins gathering across the days of spring. By late winter, many lilac buds have completed enough chilling to become capable of renewed growth. Cold weather may still hold the branches in place, but the internal barrier that maintained deep dormancy has weakened. From this point onward, development begins responding more strongly to warmth. Plant scientists often use the word forcing for the warm conditions that push a ready bud toward opening. Each mild day contributes a small amount. Several cold days may add almost nothing. A warm afternoon followed by another week of freezing weather usually produces little visible change. The tissues inside the bud respond to the pattern of temperature across time. Researchers can describe part of this pattern with growing degree days. A degree day model begins with a chosen base temperature. This base represents a level below which development is expected to proceed very slowly, or not at all for the plant being studied. The average temperature for a day is compared with that baseline. Any usable warmth above it is added to the total. Suppose a model uses a base of 5 degrees Celsius. A day with an average temperature of 9 degrees Celsius would contribute 4 degree days. Another day averaging 6 degrees would add 1 more. The running total would then reach 5. Real models may use maximum and minimum temperatures, upper limits, or other adjustments. But the central idea remains simple. Warmth accumulates. A lilac does not contain a tiny thermometer or perform this calculation. Degree days are a tool created by people to describe the effect that repeated temperatures have on biological development. Inside the bud, warmth changes the speed of chemical reactions. Enzymes become more active. Cells resume growth. Water movement increases and stored sugars become available for developing tissues. These responses build gradually rather than beginning at full strength after one mild afternoon. This helps explain why 2 lilacs in the same town may open several days apart. A shrub, beside a south-facing brick wall, receives direct sunlight and heat released from the masonry after sunset. The air and soil around it may remain warmer than conditions in an open yard nearby. A sheltered corner also loses less heat to wind. Lilacs on north-facing slopes receive less direct spring sunlight and may develop more slowly. Higher elevations are usually cooler than nearby lowlands. Latitude changes the average timing of seasonal warmth over much larger distances. A lilac in southern Ontario will generally reach its spring heat requirements before the same cultivar growing much farther north. Snow cover adds another layer. Bright snow can reflect sunlight, yet a deep layer also insulates the ground and delays soil warming. Air temperatures may rise well above freezing, while roots remain surrounded by cold soil. Near the Great Lakes, large bodies of water warm slowly during spring. Cool air moving inland can delay leafing and flowering along the shoreline compared with places farther inland. Urban areas often behave differently. Roads, buildings, and dark roofs absorb solar energy during the day and release some of it after sunset. This urban heat effect can advance plant development by several days within the same general region. Because lilacs respond visibly to these local differences, their first leaves and flowers became useful markers of seasonal change. Beginning in the 20th century, observers across North America recorded the dates when selected lilacs reached clearly defined growth stages. Some of these plants were clones, meaning they had been reproduced from the same original plant and carried nearly identical genes. One important cultivar was Syringa X chinensis, red Rothomagensis. Its records later contributed to the extended spring indices used by the USA National Phenology Network. Phenology is the study of recurring natural events, including leaf opening, flowering, migration, and insect emergence. The spring indices use temperature records and historical plant observations to estimate when early season leafing and flowering should occur across broad areas. A single lilac beside a house remains a local plant. Thousands of comparable observations can reveal the movement of spring across states, provinces, elevations, and decades. The calculation does not capture every influence. Soil moisture, plant health, shade, pruning, and genetics also affect timing. Still, accumulated warmth explains much of the steady movement from a closed winter bud toward active growth. As that total rises, the change finally becomes visible. The bud grows rounder, the outer scales loosen, and the folded flower cluster inside begins pressing into the spring air. The first visible change is a slight swelling beneath the outer scales. A bud that remains small and firm through winter begins to look rounder. Its protective scales separate at the edges as the tissues inside press outward. A narrow tip of green or red-purple may appear between them. The flower cluster is still tightly folded, with its smaller branches packed close to the central stalk. Much of its basic structure was formed before winter. Spring growth enlarges those prepared parts and completes many of the chemical changes required for flowering. Water is essential to this expansion. As frozen ground thaws, liquid water becomes available around the lilac's fine roots. Root hairs increase the surface area in contact with the soil and absorb water along with dissolved minerals. The water enters the xylem, the system of narrow tubes that runs through the roots, stems, and young shoots. Movement through the xylem is supported by several forces. Water molecules cling to one another, helping them remain connected in long columns. They also cling to the inner walls of the vessels. Evaporation from young leaves creates an upward pull, while pressure from the roots can contribute under some conditions. No single force opens the bud by itself. Water transport works together with stored energy, active metabolism, cell division, and cell expansion. Inside the bud, water crosses cell membranes and enters a large internal compartment called the vacuole. As the vacuole fills, the cell contents press outward against the firm cell wall. This force is called turgor pressure. Turgor helps keep plant tissues upright and gives expanding cells the pressure needed to increase in size. A wilted leaf has lost much of this pressure. A swelling lilac bud is gaining it. The cell wall does not stretch without control. Special proteins and enzymes loosen selected parts of the wall, allowing it to expand while maintaining its shape. Sugars stored in the shrub provide energy for these reactions. They also supply raw material for new membranes, proteins, pigments, and other compounds. The compressed panicle gradually begins to lengthen. Its central axis extends first, carrying the smaller side branches outward. Those branches separate from one another, creating the open framework that will later support many individual florets. The narrow corolla tubes grow longer. Petal lobes at their tips remain folded together for a time, then begin to separate. Research on Syringa oblata, a lilac species native to parts of China and Korea, has divided this process into clear, visible stages. The earliest stage contains a tight red-purple bud. During initial opening, the corolla tube lengthens, while the petal lobes remain partly closed. At full bloom, the four lobes spread outward until they sit at roughly right angles to the tube. Later, the petals become paler and begin curling at the edges. These stages can exist together on one panicle. Flowers near the base may already be open, while those closer to the tip remain narrow buds. This staggered development prevents the entire cluster from passing through bloom in one brief moment. Weather can stretch or compress the schedule. Cool spring days slow many chemical reactions inside the flower. Cell expansion proceeds more gradually, and individual florets may remain fresh longer. A period of sustained heat speeds development. Many buds may open close together, creating a strong display that fades sooner. Dry conditions can limit water supply, while heavy rain may weigh down panicles and damage delicate petals. The opening process therefore reflects both the structure prepared months earlier and the conditions surrounding the shrub at that moment. What seems sudden from a distance is a controlled sequence of hydration, growth, and unfolding. As the petal lobes spread into the air, their color also begins to shift. The purple visible in a tight bud does not remain fixed throughout flowering. Pigments are produced and altered as the flower matures, changing the shade from its earliest red-purple stage toward violet, pale lavender, and eventually near white. As the petals widen, their color continues changing inside living cells. Much of the purple comes from a group of water-soluble pigments called anthocyanins. These pigments are stored mainly inside vacuoles, the fluid-filled compartments that occupy much of a plant cell. Anthocyanins can produce red, purple, and blue tones in flowers, fruits, and leaves. Their visible effect depends on several conditions working together. The exact pigment molecule matters. Its concentration changes the strength of the color. Acidity inside the vacuole can shift the shade, while nearby compounds and metal ions may alter how light is absorbed and reflected. A petal therefore gains its color from a chemical environment rather than from one simple purple substance. Anthocyanins belong to a larger family of plant compounds called flavonoids. Plants build flavonoids through a chain of chemical reactions known as a metabolic pathway. Each step is controlled by an enzyme, which is a protein that helps a particular reaction happen. Genes contain the instructions used to build those enzymes. When the activity of certain genes rises or falls, the balance of pigments inside the petal can change. Researchers have followed this process closely in Syringa oblata, a lilac species native to China and Korea. Its flower buds begin with a dark red-purple appearance. As the corolla tube lengthens and the lobes start opening, the flowers become more clearly violet. At full bloom, they often appear light violet. During the fading stage, the petals grow pale and may approach white before they curl and dry. Chemical measurements taken during those stages showed that anthocyanin levels changed as the flowers developed. The shift in color was connected with changing gene activity and pigment metabolism inside the petal. Sunlight and weather may still influence fading, but the process also comes from active changes within the flower itself. Two pigment groups help explain the range of shades. Compounds derived from cyanidin often contribute red-purple and magenta tones. Pigments derived from delphinidin are more closely associated with violet and blue-violet colors. The proportions of these compounds can change as the flower opens. Other chemical modifications may add small molecular groups to a pigment, affecting its stability, location, or final appearance. Modern research can examine these changes at a scale far smaller than the petal. One major study of Syringa oblata assembled approximately 1.05 billion DNA bases across 23 chromosomes. A chromosome is a long package of DNA carrying many genes. The researchers predicted more than 35,000 protein-coding genes within the genome. Only a fraction of those genes directly control flower color. Yet the full genetic map helps scientists locate the pathways involved in pigment production and fading. Garden lilacs display the results of natural variation and human breeding. Deep violet cultivars contain strong visible pigment across much of the petal. Pink and magenta flowers reflect different pigment balances and concentrations. White lilacs generally accumulate far less visible anthocyanin in their petals. Their cells still contain many other compounds, but the pigments responsible for strong purple tones are greatly reduced or absent. Cream-colored flowers may gain a faint warm appearance from other pigments and from the natural color of the petal tissue. The cultivar primrose is often described as yellow, although its flowers usually appear pale cream or soft yellow rather than brightly golden. Bicolored lilacs can divide pigment across different areas of the same floret. Sensation, for example, carries purple petals edged with a narrow white border. That boundary forms because pigment development differs between the center and margin of each petal lobe. Cultivars sold as blue usually remain within a pale violet or blue-violet range. The word blue is useful in horticulture because it separates cooler shades from redder purples. Light conditions also influence what the eye sees. A flower photographed in warm evening light may appear more pink. The same cluster under a cool, overcast sky can look noticeably bluer. Through all these changes, lilac remains a color produced by living chemistry. Pigments are built, modified, stored, and gradually reduced as each flower moves through its short life. The name of that color is often shared with lavender, another fragrant purple flower whose similar appearance comes from a very different family, habitat, and evolutionary history. The words lilac and lavender often appear beside one another. Both can describe flowers, pale shades of purple, perfumes, candles, soaps, and fabrics. Their names also carry similar associations with spring gardens, clean linen, and quiet rooms. That overlap can make the plants seem closely related. Botanically, they belong to two separate families. The common lilac, Syringa vulgaris, belongs to Oleaceae, the olive family. This family also includes olive trees, ash trees, forsythias, privets, and jasmines. English lavender, Lavandula angustifolia, belongs to Lamiaceae, the mint family. Its relatives include rosemary, sage, thyme, basil, and the many plants commonly called mints. Members of the mint family often have strongly scented leaves and square stems. Lavender shows both features. A young lavender stem usually has 4 clear sides, and tiny oil-producing structures cover much of its foliage and flowers. Crushing a leaf between the fingers releases aromatic molecules almost immediately. Lilac carries its fragrance mainly in the flowers. Its leaves have a much milder smell, and its rounded woody stems lack the square shape common among mint relatives. The two plants also build very different bodies. A common lilac may rise 12 feet or more, with thick woody stems and a broad root system that can send up new shoots. Its leaves fall during autumn, leaving a bare framework through winter. Cold exposure becomes part of the process that prepares its flower buds for spring. English lavender stays much lower. A mature plant often forms a rounded mound between 1 and 3 feet tall, depending on the cultivar and growing conditions. Botanists describe it as a subshrub. A subshrub is a small plant with woody lower stems and softer younger growth above them. Lavender may remain evergreen or semi-evergreen, meaning that it keeps all or some of its narrow grey-green leaves through winter when conditions allow. Its leaves are adapted to bright sun and relatively dry ground. Their narrow shape limits the surface area exposed to heat, while fine hairs give many varieties a silvery appearance and help reduce water loss. The native ranges of the plants reflect these different strategies. Wild common lilac comes from southeastern Europe, extending from the northern Balkan Peninsula into northern central Romania. It evolved in a region with clear seasonal changes and cold winters. English lavender is native from northeastern Spain, across southern France toward Italy. Much of this range has hot, dry summers, rocky slopes, and soils that drain water quickly. A lilac often struggles where winter is too mild or summer heat remains intense for long periods. Lavender can suffer in heavy, wet soil because water filling the spaces around its roots reduces oxygen and encourages decay. Their shared purple color developed through separate evolutionary histories. Both use pigments that absorb some wavelengths of visible light. and reflect violet or purple tones toward the eye. Their flower shapes differ greatly. Lilac produces many four-lobed tubular florets within branching panicles. Lavender carries small two-lipped flowers along narrow spikes. The two-lipped form is common in the mint family and creates a landing surface for visiting insects. Fragrance creates another familiar resemblance, although the chemical mixtures remain distinct. Lavender essential oil is commonly rich in linalool and linaloyl acetate. Linalool has a soft floral aroma and occurs in many unrelated plants. Linaloyl acetate contributes a sweet, smooth quality that is especially important in English lavender oil. The exact proportions vary with cultivar, climate, harvest time, and the way the oil is distilled. Lilac flowers also produce linalool. Their cells transform some of it into a group of related molecules called lilac aldehydes and lilac alcohols. In chemistry, the words aldehyde and alcohol describe particular arrangements of atoms within a molecule. They do not mean that a flower contains a household cleaner or a drinkable form of alcohol. These lilac compounds contribute green, floral, and softly powdery notes that help separate the living scent of lilac from the sharper herbal character of lavender. Shared molecules create a small area of chemical overlap. The full fragrances remain different because each plant releases its own mixture in its own proportions. A lavender sprig carries scent in its leaves and flowering spikes for an extended period. A lilac concentrates much of its fragrance into a short spring display, produced by hundreds of open florets. Linalool provides one chemical connection between them, while the lilac's distinctive character emerges as flower cells reshape that starting material into several molecules with different structures and different effects on the nose. Linalool gives lilac and lavender one point of chemical overlap, though the full scent of a living lilac comes from a much larger mixture. Dozens of airborne compounds can leave the flowers at the same time. Some smell floral or green. Others add faint, waxy, spicy, or powder-like notes that may be difficult to notice alone. Together, they create the fragrance recognized within a few steps of a blooming shrub. These airborne substances are called volatile organic compounds. Volatile means that a substance can enter the air fairly easily under ordinary conditions. Organic means that its molecules are built around carbon atoms. A flower releases these compounds from its petal surfaces and other tissues, allowing them to travel beyond the plant. Linalool is one of the starting materials used in lilac scent chemistry. Inside the flower, enzymes modify its structure through several chemical steps. An enzyme is a protein that helps a particular reaction happen more quickly. During these reactions, oxygen-containing groups are added or rearranged. The products include lilac aldehydes and lilac alcohols, two groups of molecules strongly associated with the flower's characteristic scent. The words aldehyde and alcohol describe small arrangements of atoms within a molecule. They refer to chemical structures rather than household alcohol, cleaning liquid, or any substance the flower stores in a bottle-like form. Researchers have traced this pathway using isotopic labels. An isotope is a slightly different form of an element, whose atoms carry the same chemical identity, but have a different mass. Scientists supplied common lilac flowers with specially labeled linalool. The label allowed them to follow those atoms through later chemical reactions. labeled material appeared in lilac aldehydes and lilac alcohols, showing that the flower had converted the original linalool into these new compounds. The same transformation was also observed in isolated chloroplasts taken from lilac petals. Chloroplasts are small structures inside plant cells. They are best known for carrying out photosynthesis in green leaves. But chloroplast-related structures in petals can also contain parts of the machinery used to build scent compounds. The molecules produced through this pathway come in several three-dimensional forms. These forms are called stereoisomers. Two stereoisomers contain the same types of atoms, joined in the same basic order. Their atoms point in different directions through space. Much like a left hand and a right hand share the same parts, while remaining shaped as mirror images. That difference can matter greatly to the nose. Smell begins when an airborne molecule fits into an odor receptor inside the nasal passages. An odor receptor is a protein that responds to certain molecular shapes. One stereoisomer may fit a receptor more easily than another. Some forms can therefore be detected at lower concentrations, while others produce a different odor quality. Olfactometric testing examines this relationship by combining chemical analysis with human smell detection. A sample is separated into its individual compounds. Trained participants smell the material as each compound leaves the instrument. Their responses help identify which molecules contribute strongly to the scent and how easily each form can be detected. Lilac aldehydes and lilac alcohols are important parts of this mixture, yet they do not create the entire fragrance by themselves. The living flower also releases linalool and a range of other volatile compounds. Their proportions can change among species, cultivars, flower ages, and weather conditions. A newly opened floret may release a different balance from one that is already fading. This complexity makes natural lilac fragrance difficult to capture. Lilac flowers do not produce a large supply of easily collected oil in the way lavender does. Heat, solvents, and storage can also change delicate scent molecules or alter their proportions. Commercial lilac perfumes are therefore usually built as accords. An accord is a blend of several fragrance materials designed to create one recognizable overall impression. Perfumers may combine synthetic lilac aldehydes with floral, green, and powdery materials until the result suggests a fresh panicle in bloom. Even a carefully made accord remains an interpretation of the flower. On the shrub, scent production changes from hour to hour as petals mature. Temperature shifts, and individual florets open at different times. Once the volatile molecules cross the petal surface, they enter moving air. Their path now depends on evaporation, temperature, wind, and the constant mixing of the atmosphere around the flowers. A scent molecule crossing the surface of a lilac petal enters a far less orderly environment. Within the flower, enzymes and cell membranes helped control where that molecule was made and stored. Outside, its movement depends on temperature, pressure, surrounding air, and chance collisions with other molecules. The change from liquid or dissolved material into gas is called volatilization. Lilac scent compounds are volatile because they can enter the air at the ordinary temperatures found around a spring flower. Warm petal surfaces usually release these molecules more readily than cold ones. Higher temperature gives molecules more energy, making it easier for some of them to escape into the atmosphere. The strength of the fragrance still depends on the flower's biology. A warm, aging floret may release a different mixture from a newly opened flower on the same panicle. Genes control scent-producing enzymes, while flower age affects how active those chemical pathways remain. Once airborne, each molecule travels through constant motion. Air molecules strike it from many directions, sending it along an irregular path that changes countless times each second. This random spreading is called diffusion. Diffusion tends to move scent away from the high concentration close to the petals and toward areas where fewer scent molecules are present. On its own, diffusion works effectively across tiny distances. The scent from a full lilac shrub can travel much farther because the atmosphere itself is moving. Warm air near sunlit leaves and branches may rise through convection. Convection is the movement produced when warmer, less dense air rises and cooler air shifts into its place. Even a gentle breeze can carry floral compounds several feet from the plant far more quickly than diffusion alone. The resulting scent does not form a smooth, even sphere around the shrub. Moving air stretches it into a plume. Turbulence breaks that plume into curling strands and scattered pockets with different concentrations. Turbulence is the irregular, swirling motion created when air passes around branches, leaves, walls, fences, and other obstacles. A person walking along a path may suddenly notice lilac fragrance several yards before seeing the flowers. One more step may carry them into air containing very few lilac molecules. The scent can return moments later, as another narrow part of the plume moves across the path. Wind direction may carry most of the fragrance toward one side of the shrub. Someone standing upwind could smell very little, while another person downwind receives a much stronger concentration. Weather changes the plume from hour to hour. Cool morning air often slows volatilization. As sunlight warms the flowers, more compounds may enter the atmosphere, provided the petals are still actively producing them. Strong wind increases dilution by mixing the scent into a larger volume of air. That same wind can carry a small number of molecules much farther from the plant. Rain changes the pattern again. Droplets can wash compounds from floral surfaces, cool the petals, and temporarily remove some material from the nearby air. High humidity may also change how people perceive a fragrance, although the effect depends on the compounds and surrounding conditions. Distance alters both strength and balance. Some molecules leave the flower in larger quantities. Others can be detected by the human nose at extremely low concentrations. Compounds also differ in how quickly they evaporate and how long they remain airborne. The blend detected close to a panicle may therefore differ slightly from the scent encountered farther downwind. Researchers separate this complicated mixture with an instrument called a gas chromatograph. A small fragrance sample is carried through a long, narrow column by a flowing gas. Different compounds move through the column at different speeds because they interact differently with its inner coating. They emerge one after another, rather than arriving as one blended scent. A second instrument, called a mass spectrometer, can then help identify them. The molecules are given an electrical charge and broken into smaller fragments. Their masses and fragment patterns create a chemical signature that can be compared with known substances. This combination allows scientists to distinguish compounds that the nose experiences together. Measurements have shown that lilac species and cultivars can release different volatile profiles. Two purple shrubs may look similar while producing different proportions of linalool, lilac aldehydes, lilac alcohols, and other scent compounds. Their fragrance plumes also change as individual flowers age across the panicle. For a person passing nearby, this shifting structure may become one of the first noticeable signs of spring. For an insect sensitive to floral odors, the same moving strands can carry useful information toward nectar, pollen, and the small reproductive structures hidden inside each flower tube. A bee moving through the shifting scent plume eventually reaches the outer edge of the flowering panicle. From its scale, the cluster is a crowded feeding ground, made from dozens of narrow floral tubes. Many florets sit only a fraction of an inch apart, allowing the visitor to move between them with short steps or brief movements of its wings. The purple petals help make the display visible against green leaves and brown branches. Fragrance carries information beyond the reach of color, especially when the flowers are hidden behind foliage or approached from the side. Once the insect arrives, the shape of each floret guides its movements more closely. 4 spreading petal lobes form a small platform around the entrance. Below them, the corolla narrows into a tube containing nectar near its base. Nectar is a sugar-rich liquid produced by specialized tissues in the flower. Its main sugars often include sucrose, glucose, and fructose, though their proportions vary among plants and through the life of a flower. A visitor receives food energy from the nectar, while the lilac gains a chance to move pollen. Different insects approach this reward with different equipment. A bumblebee has a sturdy, hairy body that can collect loose pollen as it presses among flowers. Many butterflies use a proboscis, a long feeding tube that remains coiled beneath the head when it is not in use. When the butterfly lands, the proboscis unrolls and reaches down into the corolla tube. Some moths feed in a similar way, including species that visit flowers during evening or nighttime hours. Tongue length influences which visitors can reach nectar efficiently. Body width and feeding position affect whether the insect touches the flower's reproductive structures. Inside a common lilac floret, 2 stamens are attached to the inner wall of the corolla tube. Each stamen carries an anther where pollen develops. A grain of lilac pollen is microscopic, but thousands can be released from the anthers across a panicle. Its outer wall protects the cells inside as the grain is exposed to air and carried between flowers. The pistil occupies the center of the floret. At its upper end is the stigma, a surface adapted to receive pollen. A visitor reaching into the tube may brush against the anthers and collect pollen on its head, mouthparts, legs, or body hairs. A later visit to another flower can bring some of those grains into contact with the stigma. This movement is pollination. The tight arrangement of a panicle makes repeated visits easier. After feeding from one floret, an insect can turn toward another opening only a short distance away. A single cluster may contain flowers at different stages, with some freshly opened and others already fading. The available nectar and pollen therefore change as the visitor moves along the branches of the panicle. Color and fragrance help bring the insect into the area. flower shape, then influences how its body meets the anthers and stigma. The reward encourages it to continue visiting. Each species interacts with the lilac differently. A broad-bodied bee may make frequent contact with reproductive surfaces. A butterfly can reach nectar while touching less pollen, depending on its posture and the length of its proboscis. A small insect may enter the opening without carrying pollen effectively between separate plants. Some animals visit only to drink nectar. Others rest on the cluster or search for prey among the flowers. Seeing an insect on a lilac therefore confirms visitation, while effective pollination depends on where it touched the flower and what it did afterward. Lilacs also support insects beyond the flowering period. The caterpillars of several large moths may feed on lilac leaves. These include the wavy-lined emerald and some sphinx moths, whose larvae consume foliage before changing into winged adults. Leaf feeding by a caterpillar is separate from the pollen transfer performed by an adult flower visitor. Both relationships place the shrub inside a wider community of insects using different parts of the plant at different stages of life. When compatible pollen reaches a receptive stigma, the visible exchange at the flower's surface is complete. The grain then absorbs moisture and begins growing through the pistil, carrying the reproductive process inward toward ovules protected inside the ovary. Petals may soon fade, while this new growth continues through tissues that will eventually become seeds and a dry brown capsule. A compatible pollen grain resting on the stigma begins by absorbing moisture from the flower's surface. Its outer wall stays in place while an inner layer pushes outward through a small opening. This new growth becomes a pollen tube. The tube is only 1 cell wide, yet it can extend far beyond the original pollen grain. It enters the stigma and grows downward through the style, the narrow stalk connecting the top of the pistil to the ovary. Chemical signals within the flower help guide its direction. Cells along the root provide water and nutrients as the tube continues growing. 2 sperm cells travel inside it toward the ovules held within the ovary. Pollination occurred when the pollen first reached the stigma. Fertilization takes place later, after the pollen tube reaches an ovule and releases the sperm cells. 1 sperm cell joins with the egg cell, producing the first cell of a new lilac embryo. The second joins with other nuclei inside the ovule and helps form a food storage tissue called endosperm. This process is known as double fertilization and occurs in flowering plants. The young embryo will eventually contain the early forms of a root and shoot. Endosperm supplies stored nutrients that support the seed during its first stages of development. Once fertilization has occurred, the flower begins redirecting its resources. Producing strong fragrance requires energy and specialized enzymes. Nectar also contains sugars that could be used elsewhere in the plant. As the value of attracting new visitors declines, scent production slows and nectar becomes less abundant. Petal cells begin aging. Their membranes weaken, pigments break down, and water pressure falls. The once firm lobes soften and curl before separating from the flower. Meanwhile, activity increases inside the ovary. The fertilized ovules enlarge as embryos develop within them. The ovary wall thickens and becomes the outer wall of a fruit. In common lilac, this fruit develops into a dry capsule. A capsule is a fruit that becomes firm and opens when its seeds are mature. Lilac capsules are usually flattened, and may measure around 3/4 of an inch long or less. Young capsules begin green because their outer tissues still contain chlorophyll. They may perform a small amount of photosynthesis, while the seeds inside are developing. Through summer, the capsule gradually loses moisture. Its color changes from green to tan and then brown. The fruit is divided internally into 2 sections. Each section can contain as many as 2 seeds, giving one capsule a possible total of 4. Some ovules may fail to develop, so many fruits contain fewer than that maximum. When the capsule reaches maturity, tension builds within its drying walls. The 2 sides eventually split apart along natural seams. Botanists describe this kind of fruit as dehiscent, meaning it opens by itself along a defined line. The opening exposes the seeds and allows them to fall or be shaken free by wind and movement of the branch. Lilac seeds are light enough to land near the parent shrub, though some may be carried farther across hard ground or crusted snow. Those that reach suitable soil still face a difficult beginning. Moisture must enter the seed. The embryo must survive winter conditions and begin growth at an appropriate time. Young seedlings also compete with grass, fallen leaves, and the roots of established plants. Many seeds never become mature shrubs. The brown capsules can remain on the panicles after their seeds have been released. Some persist into autumn or winter, standing where the purple flowers had opened months earlier. Gardeners often remove faded panicles soon after flowering. This practice improves the shrub's appearance and prevents energy from being directed into mature fruit and seed. Removing old flowers does not guarantee a heavier bloom the following year. Flower production also depends on summer growth, available sunlight, pruning time, winter chilling, weather, cultivar, and the general condition of the plant. A lilac can therefore produce fewer flowers after a difficult season, even when every faded panicle was removed. Seeds remain important because they combine genetic material from 2 parents. Each seedling may differ in color, height, fragrance, flowering time, or resistance to disease. That variation gives lilacs a way to change across generations. An established shrub can also continue without producing a seed at all. Beneath the soil, connected roots and underground stems may send up new shoots, carrying the same genetic identity as the older plant above them. Close to the parent shrub, a different kind of new growth may rise through the soil. These young stems are called basal shoots when they emerge near the crown, the area where the roots meet the stems. Shoots appearing farther away are often called suckers. They can grow from roots or other connected tissues beneath the ground. Common lilac naturally has a suckering, multi-stemmed growth habit. A well-established plant may begin with only a few upright stems and gradually widen as new shoots appear around the original base. Given enough time, the planting can develop into a dense colony several feet across. The shoots are produced through vegetative reproduction. Vegetative reproduction creates a new stem from the tissues of an existing plant, without first producing a seed. Cells divide through a process called mitosis, which normally passes the same genetic information into the new growth. A sucker growing from an own root lilac therefore carries essentially the same genome as the shrub that produced it. Its flower color, general growth form, and many other inherited traits should match the parent. A seedling develops differently. Its genes come from an egg cell and pollen, so it receives a newly mixed set of genetic instructions. The resulting plant may differ from both parents. A sucker continues the existing genetic individual into another stem. This changes how the age of an old lilac should be understood. One thick branch may survive for several decades, but it does not need to remain forever. Younger stems can rise beside it while sharing the same connected root system. A mature shrub may contain smooth shoots, only a year or two old beside trunks with rough bark and much greater age. The visible bush becomes a collection of living modules. A module is one repeated part of an organism that can grow, branch, and eventually be replaced. Roots support many stems. Each stem carries its own branches and leaves, while contributing sugars to the larger connected system. When an old stem weakens, younger growth can take over part of its space. The common lilac's persistence, therefore, comes partly from renewal. An individual trunk may decline, while the clonal planting continues through stems produced later. Older branches often become tall and crowded. Their lower portions may lose leaves as the canopy above blocks sunlight. Flowering can become concentrated near the highest tips, where the branch still receives strong light. Weak wood may also break under snow or strong wind. Gradual renewal pruning helps shift the shrub toward younger growth. The Arnold Arboretum recommends removing roughly 1/3 of the oldest stems at ground level each year, for about 3 years. Cutting near the base opens space within the shrub and allows more sunlight to reach younger shoots. The remaining older stems continue carrying some flowers, while the replacement growth develops. Removing every stem at once is a more severe form of renovation. A healthy root system may respond by producing vigorous new shoots, sometimes several feet tall during a growing season. Those shoots mainly invest in leaves and wood at first. Because lilacs prepare flowers on growth formed during an earlier season, a heavily renovated plant may need 1 or more years before it produces a full display again. Propagation history also affects which shoots should be kept. Many lilacs grow on their own roots, meaning every stem and sucker belongs to the named plant. Other lilacs have been grafted. Grafting joins the upper part of one plant to the rooted lower portion of another. The selected upper piece is called the scion. The supporting rooted plant is called the rootstock. A breeder may graft a desired cultivar onto a vigorous rootstock to reproduce it more quickly or support its early growth. The tissues unite at a visible graft line near the lower stem. Any sucker arising from below that junction may belong to the rootstock. It can grow strongly while producing flowers that differ from those of the named cultivar above it. A double white lilac, for example, could be joined to a rootstock with smaller purple flowers. Allowing rootstock suckers to dominate may gradually change the visible character of the shrub. Own root plants avoid that particular problem because their underground shoots remain genetically matched to the flowering stems above. Through roots, crowns, suckers, and replacement stems, a lilac can occupy the same patch of ground long after its earliest branches have disappeared. This multi-stem form represents only one design within the lilac genus. Across Europe and Asia, related species have developed into compact shrubs, unusual leaf forms, and small trees shaped by very different climates. Beyond the garden, the lilac genus stretches across a broad band of Eurasia. The common lilac, Syringa vulgaris, occupies the western edge of that range. Its wild populations come from the northern Balkan Peninsula and extend into northern central Romania. Rocky slopes, open woodland edges, and seasonal mountain climates helped shape a shrub able to survive cold winters and flower after spring warmth returns. Most other recognized lilac species are native farther east. The genus Syringa reaches from southeastern Europe through the Himalayas, across large parts of China, and onward into Korea, the Russian Far East, and Japan. Taxonomists sometimes disagree about where one species ends and another begins. New genetic evidence can also change how populations are classified. For that reason, the exact number of accepted lilac species may shift between botanical references. The geographic pattern remains clear. Lilacs form a mainly Asian lineage with one especially familiar European member. Syringa oblata grows naturally across northern and central China and into Korea. Its name refers to the broad, rounded form of its leaves. Some populations flower earlier than common lilac, making the species valuable to breeders seeking plants that open near the beginning of the lilac season. Its flowers may pass from red-purple buds into violet bloom before fading toward a much paler shade. Farther southwest, Syringa emodi grows along the Himalayan region, from Pakistan through Nepal and into Tibet. It is often called Himalayan lilac. This species can become a large shrub or small tree, sometimes reaching around 15 feet or more. Its native environments include mountain valleys and slopes. Where winters are cold, sunlight can be intense, and growing seasons vary sharply with elevation. The leaves are longer and narrower than those of common lilac. Its flowers often appear creamy white or faintly pink, and are carried in long panicles. The Japanese tree lilac, Syringa reticulata, represents another major change in form. It grows naturally in parts of East Asia, including Japan and regions of the Russian Far East. Instead of remaining a broad thicket of many similar stems, it can develop into a small tree with one main trunk, or several prominent trunks. Mature specimens may reach 20 to 30 feet tall. Its creamy white flowers open later than those of common lilac, often near the beginning of summer. The bark can become reddish-brown and marked with pale horizontal lines, giving the tree a distinctive appearance even outside its flowering period. Leaf form also varies across the genus. Most lilacs carry simple leaves, meaning each leaf has one undivided blade. Some Afghan lilac forms produce blades cut into deep, narrow sections. The divisions can make the foliage appear delicate and almost fern-like, while each leaf remains one connected structure. Syringa pinnatifolia is even more unusual. Its leaves are compound, meaning one leaf is divided into several separate leaflets attached along a central stalk. This trait is rare enough within Syringa to make the species easy to distinguish from most of its relatives. These differences developed as lilac populations remain separated across great distances. A mountain range can limit the movement of pollen and seeds between valleys. Dry plateaus favor different traits from moist woodland margins. Long winters place pressure on buds and stems, while shorter winters alter the timing of dormancy. Pathogens and leaf-feeding insects also vary from one region to another. Across many generations, individuals with useful inherited traits leave more descendants in their local environment. A population exposed to severe cold may gradually become more cold-hardy. Plants growing under shorter spring seasons may benefit from earlier flowering. In a region with different fungal diseases, resistance can become more common. This slow process produces divergence, the gradual development of differences among related populations. The lilac genus now includes low compact shrubs, broad suckering colonies, and small trees. Some carry familiar heart-shaped leaves, while others produce narrow blades or deeply divided forms. Their flowers open at different points from early spring into early summer. A collection containing several lilac species, therefore, does not bloom as one synchronized mass. Early forms can begin opening while common lilacs are still tightly budded, and later tree lilacs may reach full flower after the earlier purple panicles have already faded. A lilac collection can pass its flowers from one plant to another across much of the spring. The earliest members begin while many common lilacs are still holding closed buds. Among them is Syringa oblata subspecies dilatata, a Korean form sometimes called early lilac. Its flower buds can open before those of Syringa vulgaris, especially when both grow under the same local conditions. Early hybrids carrying genes from Syringa oblata may follow a similar schedule. Their inherited response to winter chilling and spring warmth allows development to begin after a different combination of temperatures. Flowering time is an example of phenology, the timing of recurring events in a living organism. Each lilac has its own inherited pattern, which is then adjusted by weather, sunlight, soil temperature, and the condition of the plant. A cold April can hold early and mid-season varieties close together. Steady warmth may spread their opening dates farther apart or push several plants rapidly into bloom. Common lilacs usually provide the central peak. During this period, traditional purple, white, pink, and double-flowered cultivars may open within days of one another. Their panicles create the display most people recognize as lilac season. Even within Syringa vulgaris, cultivated varieties can differ by more than a week. One may already be fading, while another nearby still carries tight-colored buds. Dwarf Korean lilac, often sold as the cultivar palabin, adds a smaller form to this sequence. A mature palabin commonly reaches about 3 to 4 feet tall and a similar width. Its compact size allows the entire rounded shrub to become covered with small lavender-pink clusters. The plant often flowers slightly later than many traditional common lilacs, extending the display without requiring the space of a 12-foot shrub. Later species carry flowering toward early summer. Preston lilacs, developed from crosses involving Asian species, usually open after common lilacs. Their leaves are often longer and less heart-shaped, while their flower clusters can appear in pink, lavender, or white. Japanese tree lilac continues the sequence several weeks later. Its creamy white panicles may appear when the familiar purple common lilac flowers have already turned brown. A mature Japanese tree lilac can reach 20 to 30 feet tall, placing its flowers above the level of most shrub lilacs. Its scent also differs. Often described as heavier and less sweet, than that of common lilac. Botanical collections use these differences deliberately. At the Arnold Arboretum in Boston, more than 400 lilac plants represent over 170 species, hybrids, cultivars, and other named forms. Together, they create a flowering season lasting approximately 5 weeks. An early Korean lilac may begin the sequence. Common and French hybrid lilacs fill the middle. Late Asian shrubs and tree lilacs carry flowers toward the end. The collection therefore shows lilac bloom as a relay rather than one brief event. Genetics determines much of the order, while each year's weather changes the spacing between the plants. Breeders have also developed lilacs capable of producing a second, smaller display. Cultivars in the bloomerang group flower heavily during spring and can form additional buds on newer growth. After the first bloom, the shrub produces fresh shoots. Some of those shoots develop flower buds quickly enough to open during summer or early autumn. This ability differs from the usual common lilac pattern, in which flowers are prepared mainly on wood formed during the previous year. The later display depends on growing conditions. Strong sunlight, adequate water, and healthy new growth support reblooming. Heat, drought, shade, or stress can reduce it. The second bloom also tends to be lighter than the spring display, with scattered clusters appearing across the shrub, rather than every branch flowering at once. Even so, it changes the plant's seasonal schedule in a noticeable way. A lilac, once expected to flower for a few spring weeks, can produce fresh color months later. Across species, hybrids, and selected cultivars, lilac season can stretch from early spring towards summer, with occasional flowers returning near autumn. These differences began as natural variation in flowering time, height, fragrance, and flower form. Breeders learned to combine those traits, test thousands of seedlings, and preserve the rare plants whose new combinations were worth carrying forward. Among thousands of lilac seedlings, a breeder may notice one plant that opens earlier, carries fuller flowers, or produces an unusual shade. That plant can become a cultivar, which is a cultivated variety selected and maintained because people value particular traits. The name of a cultivar is usually written inside single quotation marks, such as primrose or sensation. A useful seedling may offer larger panicles, stronger fragrance, compact growth, or better resistance to disease. Its value comes from the combination of genes it inherited. Breeders cannot simply instruct a lilac to produce white-edged petals or double flowers. They create genetic variation, grow the resulting plants, and search for rare combinations worth preserving. One method is controlled hybridization. A breeder chooses two parent plants whose traits may work well together. Pollen is collected from the anthers of one parent and placed onto the receptive stigma of the other. The resulting seeds contain genetic material from both plants. During the formation of pollen and egg cells, chromosomes are rearranged and divided. Fertilization combines one set from each parent, producing a new mixture of genes in every seed. A breeder may need to raise hundreds or thousands of seedlings before finding one exceptional plant. Lilacs also grow slowly enough that evaluation can take years. A young seedling must become large enough to flower. Its first bloom may not reveal its final mature form. The breeder may continue watching it through several seasons, to judge flower color, panicle size, plant shape, and yearly reliability. A seed collected from a named lilac does not produce a guaranteed copy of that cultivar. The seedling receives a new genetic combination and may return to smaller flowers, a different color, or a less desirable growth habit. Once a valuable plant has been found, it must be reproduced vegetatively. Vegetative propagation creates new plants from existing tissue while preserving the selected genome. Cuttings can be taken from young shoots and encouraged to form roots. Division separates rooted shoots from an established plant. Grafting joins a piece of the selected cultivar onto a compatible rootstock. Modern nurseries may also use tissue culture, in which tiny pieces of plant tissue are multiplied under sterile conditions. These methods allow thousands of gardens to receive genetically matching descendants of one original seedling. Much of modern lilac breeding grew from work carried out in Nancy, France. During the late 19th century, the Lemoine nursery introduced many influential cultivars, including forms with larger panicles and additional petals. Victor Lemoine and later members of his family repeatedly crossed selected lilacs and evaluated their offspring. The term French hybrid eventually became a broad nursery label for many improved common lilac cultivars. Even though not every plant sold under that description came directly from the Lemoine family. The double white cultivar, Madame Lemoine, shows how far selection altered the wild flower form. Its florets contain extra petal-like parts, giving the panicle a dense, layered appearance. Other breeders continued exploring color. Sensation produces purple petal lobes edged with narrow white margins. Primrose opens with a pale creamy yellow cast, an unusual tone among lilacs dominated by white, pink, and purple. A more recent breeding story began in 1978 at the Arnold Arboretum in Boston. Researchers requested seed from the Chinese Botanical Academy in Beijing. The seed arrived in 1979 and produced a group of genetically different seedlings. One plant eventually stood out because it carried flowers along much more of each branch than a typical lilac. Its panicles appeared near the branch tips and at many points farther down the stems. Years of observation confirmed that this flowering pattern remained stable. The Arnold Arboretum officially introduced the cultivar Lilac Sunday in 1997. A flowering branch can create a display more than 2 feet long. Every plant sold under that name descends vegetatively from the selected original, preserving the same unusual arrangement. Cultivars such as these record decades of crossing, waiting, comparison, and propagation. Their inherited traits expand what a lilac can look like and when it can flower. Their daily survival still depends on the same physical conditions as any wild shrub. Sunlight must reach the leaves. Roots need water and oxygen. Pruning must respect the hidden flower buds. And the plant must continue sharing its tissues with fungi and insects in the surrounding garden. Once a selected lilac leaves the nursery, its inherited traits meet the conditions of an ordinary yard. A cultivar may carry genes for large panicles or unusual color, yet those traits require enough energy to become fully visible. Sunlight supplies most of that energy. Lilacs generally flower best when their leaves receive at least 6 hours of direct sun each day. In deep shade, the shrub may remain alive and continue producing green shoots. Flowering usually becomes lighter because the leaves manufacture less sugar through photosynthesis. The plant directs its limited resources toward basic growth and maintenance, leaving less available for the many flower buds prepared during summer. Shade can also keep leaves cooler and damp for longer periods after rain or morning dew. These conditions may favor powdery mildew, a group of fungi that produces a pale grey or white coating across leaf surfaces. The fungus draws nutrients from living cells beneath the coating. On an established lilac, powdery mildew is often more damaging to appearance than to survival. A heavily affected plant may still return the following year, though repeated stress can reduce its strength. Below the soil, the roots face a different balance. Lilacs prefer ground that remains moderately moist, while allowing excess water to drain away. Healthy soil contains small spaces between particles. Some spaces hold water, while others contain air. Lilac roots use oxygen from those air pockets for cellular respiration, the process that releases usable energy from sugars. Persistently saturated soil fills most of the spaces with water. Oxygen then moves toward the roots much more slowly. Fine roots may weaken or die, reducing the plant's ability to absorb water and minerals even while it stands in wet ground. Heavy clay can create this problem where water drains poorly. A low area that collects runoff may also remain saturated after storms. Raised planting sites and soils containing stable organic matter can improve drainage, though adding a small pocket of loose soil inside dense clay may simply create a basin that holds water around the roots. Water supply also affects new growth. A newly planted lilac needs regular moisture while its roots spread into the surrounding ground. An established shrub can withstand some dry periods, but severe drought may limit shoot growth and reduce the number of strong buds formed for the following year. Fertilizer cannot correct every weak bloom. Nitrogen encourages leaves and stems. An excessive supply can produce long green shoots while flowering remains sparse. Shade, pruning time, winter conditions, root health, and cultivar genetics may all be more important than the amount of fertilizer applied. Pruning creates one of the clearest examples of timing controlling the result. A lilac cut back soon after flowering still has much of summer to produce new shoots and begin forming future buds. The same cuts made in winter can remove those prepared buds from an otherwise healthy plant. Spring may then bring vigorous leaves without the expected panicles. The shrub has retained its ability to grow, while an entire season of flowers disappeared with the clipped branch tips. Crowded stems create additional pressure. Dense foliage slows air movement through the center of the shrub. Moisture remains on leaves longer, giving fungal spores more opportunity to germinate. Removing selected older stems can improve airflow and bring sunlight toward younger growth. Cleaning up badly infected leaves may reduce some material that could carry disease into another season. These practices lower risk without creating complete protection. Lilacs can also develop bacterial blight. Cool, wet spring weather may favor the bacteria responsible for blackened shoots, damaged buds, and dark patches on leaves. Leaf spot diseases create smaller areas of dead tissue, often surrounded by darker margins. Some insects use the lilac more directly as food. Scale insects attach to stems and feed through narrow mouthparts, while protected beneath waxy or shell-like coverings. A heavy population can weaken twigs by repeatedly removing plant fluids. Lilac borer larvae enter the wood. The young insect tunnels beneath the bark and through internal tissue, disrupting the movement of water and sugars. Affected stems may wilt, crack, or produce sawdust-like material near an opening. Older or stressed branches are often more vulnerable than vigorous young growth. The garden lilac therefore lives within a network of sunlight, soil organisms, weather, and competing forms of life. Its woody structure gives it considerable resilience, though every flowering season still reflects the conditions surrounding it. In colder northern landscapes where those conditions suited the plant, lilacs sometimes survived with very little care. Their roots continued producing stems beside old foundations, even after paths vanished and the houses they once bordered were gone. Those surviving shrubs sometimes became the clearest sign that a home had once stood nearby. A lilac thicket beside a stone foundation could remain after wooden walls decayed, paths filled with grass, and field edges returned to forest. Common lilac originated in southeastern Europe and reached North America through cultivation. Settlers valued the shrub because it tolerated cold winters, returned reliably from established roots, and could be shared through suckers or cuttings. A neighbor did not always need to grow a lilac from seed. A rooted shoot could be separated from an older planting and moved to another property, preserving the same genetic individual in a new location. This method helped lilacs spread from gardens near ports and towns into farms across colder parts of the continent. Some early plantings have become historical landmarks. Lilacs growing at the Governor John Wentworth Estate in Portsmouth, New Hampshire, are believed to date to approximately 1750. The word believed matters because a living shrub does not carry a planting label from the 18th century. Its age must be reconstructed from property records, local accounts, and the history of the garden. Thomas Jefferson left clearer written evidence. In 1767, he recorded a method for planting lilacs at Shadwell, his family property in Virginia. Jefferson later developed extensive gardens at Monticello, where he kept detailed notes about plants, weather, and seasonal work. George Washington also recorded lilacs at Mount Vernon. His diary entry for March 3rd, 1785, mentions transplanting lilacs and other flowering shrubs into the garden. These notes show that lilacs were already established features of prominent North American landscapes before the end of the 18th century. Their movement continued through smaller gardens whose records were rarely preserved. One especially important planting began in Massachusetts after Benjamin Bussey acquired a large property in 1806. Bussey was a merchant and farmer who developed the land into an estate later known as Woodland Hill. He is believed to have planted lilac hedgerows there soon after his arrival. The property eventually became part of Harvard University's Arnold Arboretum, founded in 1872. By then, portions of Bussey's old lilac plantings had survived for decades. Some original stems disappeared as individual wood aged and died. Younger suckers continued rising from connected roots. The Arnold Arboretum later propagated surviving material and recreated parts of the historic hedges, allowing the planting pattern to continue even where the earliest stems were gone. This survival reflects the lilac's modular growth. A wooden house depends on walls and beams remaining connected in one structure. A lilac colony can lose individual stems while new ones rise from the same root system. Cold hardiness protects dormant buds through northern winters. Stored carbohydrates support fresh shoots after damage or neglect. Suckering allows the occupied patch of ground to widen slowly across the years. The shrub's persistence also helped turn lilac flowering into a public tradition. The Arnold Arboretum established Lilac Sunday in 1908. Visitors gathered during the bloom period to walk among the collection and compare flowers differing in color, fragrance, size, and form. Attendance grew as the event became associated with spring in Boston. A reported 43,000 people visited for Lilac Sunday in 1941. The exact crowd belonged to one day, while the plants had been developing toward it since the previous summer. Their buds passed through autumn dormancy, winter chilling, and the uneven warmth of a Massachusetts spring before the gathering could take place. The same biological cycle continued after the visitors returned home. Old garden lilacs do not literally hold memories of the families who planted them. Their living tissues preserve continuity through ordinary processes involving roots, cell division, stored energy, and replacement growth. That biology can still create a powerful historical marker. A shrub may remain beside a cellar hole after the roof has vanished. Flowers may open along a former fence line whose wood has completely decayed. The plant shows where care was once repeated over many years, because pruning, transplanting, and protecting young shoots helped establish the colony that later survived alone. Repeated flowering also provides something more precise than a general connection with the past. The date of the first open leaf can be written down. The first fully open flowers can be recorded each spring. When those observations continue across many years and locations, an old garden shrub becomes part of a scientific record, measuring how the seasons themselves are changing. To make that record useful, observers must describe the same stage in the same way. A note saying that spring felt early provides a personal impression. A dated observation of the first open flower gives researchers something that can be compared across years and locations. The study of recurring biological events is called phenology. Phenology includes the timing of leaf opening, flowering, insect emergence, bird migration, and fruit ripening. Researchers examine how these events respond to temperature, rainfall, daylight, elevation, and other environmental conditions. Lilacs are especially useful for this work because their seasonal changes are easy to recognize. A dormant branch begins with closed buds. Later, the leaf tips become visible between separating scales. A flower bud expands into a colored cluster. Eventually, individual florets open far enough for their reproductive parts to function. Each stage can be described with a clear definition and assigned a date. Lilacs also remain in one location for many years. The same shrub can be observed each spring without needing to find a new plant. Common lilacs grow across large parts of North America, especially in regions with cold winters. Their widespread cultivation allows volunteers in distant towns and climates to record comparable events. Genetic variation creates a challenge. Two lilacs growing beside one another may open on different days because they belong to different cultivars. One may require more winter chilling. Another may respond more quickly to spring warmth. Researchers reduce this source of variation by distributing cloned plants. A clone is a plant reproduced from the tissue of one original individual. Clone plants carry nearly identical genetic instructions, so differences in their flowering dates are more strongly connected to local environmental conditions. One important plant used in North American monitoring is Syringa X chinensis, red Rothamgensis. It is commonly called a cloned lilac within the observation program. Matching plants were established at many sites, allowing researchers to compare how the same genotype responded in different climates. A clone in a warm city might open before one growing at a higher elevation. Another near a cold lake shore might remain closed longer, even at a similar latitude. Historical lilac and honeysuckle observations began accumulating during the 1950s. Teachers, gardeners, naturalists, and other volunteers recorded seasonal stages on paper forms and sent their results to coordinating scientists. Modern observers can enter similar information through programs such as Nature's Notebook, operated by the USA National Phenology Network. The observation itself remains simple. The recorder identifies the plant, checks a defined life stage, notes the date, and provides the location. Consistency gives those small records their value. Researchers must also check the data for possible mistakes. A flowering date may be entered under the wrong year. The observer may have identified a different lilac cultivar. An unusually late record could represent a second bloom rather than the main spring flowering period. Local weather records help scientists decide whether an observation makes environmental sense. One study examined common and cloned lilac flowering records, collected from 1980 through 2013. Its screening process judged approximately 97% of the observations to be environmentally consistent. That high percentage showed that volunteer records could provide dependable scientific information when the instructions were clear and the results were carefully checked. The remaining records still mattered because removing them changed the calculated long-term trend. After inconsistent observations were excluded, the estimated shift in flowering time differed by about 2 days per decade from the result produced by the original dataset. This does not mean that every lilac advanced by exactly 2 days. It shows how a relatively small number of questionable records can alter a conclusion drawn across several decades. Quality control becomes especially important when scientists use plant observations to study climate. Lilac and honeysuckle records help create the extended spring indices. These mathematical models estimate the timing of first leaf and first bloom using daily temperature information. Researchers can map the results across large areas, showing spring development moving northward and into higher elevations. The models support work involving agriculture, wildfire conditions, water cycles, and seasonal allergies. A flowering shrub beside one home contributes only one date. Thousands of carefully recorded dates form a network extending across much of a continent. Together, they show that the lilac's schedule changes with geography and from one year to another. Across several decades, some springs stand apart sharply, with leaves and flowers opening weeks earlier than people in the same locations had come to expect. The long record becomes especially revealing when one unusually warm year is placed beside the decades that came before it. Spring temperatures control how quickly usable warmth accumulates after dormancy has been released. When mild weather arrives early and remains in place, leaf buds can begin expanding sooner. Flower development may also advance, sometimes by several weeks compared with a colder year at the same location. The year 2012 provided a striking example across much of North America. In the lilac monitoring record, cloned plants averaged breaking their leaf buds on March 19th. That was the earliest average in a record, then extending back to 1961. The average first report of open flowers arrived on April 20th, which was also the earliest flowering onset recorded for those cloned lilacs. Common lilacs followed a similar pattern. Their leaf buds averaged opening on March 28th. Their first open flowers were reported, on average, by April 22nd. These dates came from many plants spread across different locations, rather than from one shrub experiencing one unusual week. Even so, averages require careful interpretation. The number and location of observers can change from year to year. A season with many reports from warmer southern areas may appear earlier than one, with more observations from colder northern sites. The USA National Phenology Network specifically warned that uneven reporting could affect the 2012 averages. Temperature records, plant identity, elevation, and local weather must therefore be examined alongside the bloom dates. The basic spring response remains clear. Warm conditions speed the chemical reactions and cell expansion that drive bud development. The winter side of the process adds more complexity. Lilacs require enough cold exposure to release deep dormancy. If winter becomes too mild, some cold-dependent cultivars may complete that requirement slowly or unevenly. A warm spring following inadequate chilling could produce buds that open at different times across the same shrub. Some may remain closed longer, while others begin developing as soon as sufficient warmth arrives. The outcome depends on the inherited requirements of the cultivar and the exact sequence of temperatures it experienced. Earlier growth also changes the plant's exposure to frost. A closed winter bud protects its flower tissues behind firm scales. Once the bud swells and the petals begin extending, those water-rich cells become less resistant to freezing. A temperature that caused little damage in January may injure an expanding shoot in April. Late frost can blacken young leaves, damage flower tips, or destroy an entire panicle shortly before opening. This creates a seasonal trade-off. Early warmth allows the shrub to begin growing sooner. That same head start places tender tissue outside its winter protection for a longer period before the final damaging freeze has passed. Local conditions can change the risk within a small area. A lilac beside a heat-holding brick wall may open before one in an exposed field. Cold air can collect in low ground during a clear night. Snow cover may keep roots and lower buds cooler, even while daytime air becomes mild. Moisture also matters because drought can limit expansion after warm temperatures have already encouraged development. Changes in plant timing can extend beyond the shrub itself. Insects respond to temperature, daylight, rainfall, and their own inherited schedules. A bee may become active during an early warm spell, while another species remains in a sheltered stage for several more weeks. Flowering earlier can alter when nectar and pollen become available. It may also shift the period when airborne pollen affects seasonal allergies. Fungi and leaf-feeding insects respond to changing warmth and moisture in their own ways. Birds that feed on spring insects may follow still different signals during migration. These relationships do not all move at the same rate, and every early season does not create a harmful mismatch. The possibility grows when connected organisms respond to separate environmental cues. Long-term trends remain difficult to express with one number. Removing a small group of environmentally inconsistent lilac records changed one estimated flowering trend by about 2 days per decade. That result showed how strongly data screening can affect the apparent rate of change. It did not mean that every lilac across North America was advancing by exactly 2 days. Cultivar, latitude, elevation, urban heat, winter chilling, and observer coverage all influence the pattern. Lilacs may also produce scattered flowers in late summer or autumn. Some modern cultivars were selected for reblooming. Other plants may respond to unusual weather, drought followed by rain, or interrupted dormancy. A second bloom, therefore, has several possible explanations and cannot be treated as one universal sign of climate change. The seasonal calendar still operates through recognizable biological rules. Those rules now interact with temperature patterns that vary more sharply from year to year. Inside one open cluster, the result remains physical and immediate. Its flowers reflect the summer when their buds first formed, the cold that altered their dormancy, and the exact sequence of spring warmth that finally brought them into the air. A single lilac panicle now carries the entire journey within its branching form. Each floret opens through 4 spreading lobes, with a narrow corolla tube leading inward toward the stamens and pistil. Purple color rests inside living petal cells, where water-soluble anthocyanins absorb part of the light and reflect violet tones toward the eye. Their balance changes as the flower matures. A tight bud may begin red-purple, an open flower may appear violet, and an aging petal can fade toward a much paler shade. Along the moist surface of the corolla, volatile molecules continue entering the air. Linalool made within the flower is transformed through several chemical steps into characteristic lilac aldehydes and lilac alcohols. These compounds mix with many others before wind carries them away in thin, shifting plumes. Water arriving through the xylem keeps the flower cells firm. Sugars moving through the phloem support scent production, petal maintenance, and the reproductive tissues hidden inside each tube. The full cluster depends on a woody framework extending downward through branches, older stems, and roots anchored beneath the soil. Its brief spring display began many months before the petals appeared. During the previous summer, growing points on young shoots changed their development and began organizing future panicles. Small floral parts formed inside buds only a few millimeters long. Protective scales enclosed them as autumn arrived. The leaves yellowed and fell, while those compact flowers remained alive on bare branches. Deep dormancy then restrained their growth through early winter. Accumulated chilling gradually altered that internal condition. Once the buds became capable of renewed development, spring warmth began controlling the pace. Mild days added usable heat across time. Cells absorbed water, tissues expanded, and the compressed branches of each panicle slowly unfolded. The flower, therefore, carries evidence from more than one season. Its structure records summer growth. Its opening depends on winter cold and the particular sequence of warmth that followed. Even the date of first bloom can become a measurement when an observer writes it down and compares it with records from other years. Within the open cluster, an insect may settle near the entrance of a corolla tube. A bee or butterfly reaching toward nectar can brush against the two stamens and carry pollen toward another stigma. Compatible pollen may then grow a tube through the style toward the ovary. Fertilized ovules can become seeds inside a dry capsule that later turns brown and splits along its seams. Each seed carries a new mixture of genes. A seedling may differ from its parents in height, color, fragrance, or flowering time. Close to the older stems, a sucker follows a different path. It rises from connected underground tissue and preserves the existing genetic identity of the shrub. One root creates variation. The other allows the established lilac to continue through replacement stems as older wood weakens and disappears. Human selection joins these natural processes. A breeder notices an unusual seedling, watches it through several flowering seasons, and preserves its traits through cuttings, grafting, or another form of vegetative propagation. That care can carry one rare combination into thousands of gardens. The same biological persistence explains the lilac hedge beside an old foundation. Roots remain active beneath changing ground. New stems rise where earlier trunks have died. Flowers may return long after the fence line has vanished and the building nearby has fallen away. Some of those blooms become part of a longer record. A dated note describing the first open flower can join observations gathered across towns, elevations, and decades. The shrub beside one home then contributes to a larger picture of spring moving through a region. Lilac and lavender often share the same pale purple imagery, though their biology remains distinct. Lilac belongs to the olive family and prepares its spring flowers through a cold winter. Lavender belongs to the mint family and carries aromatic oils through a much smaller plant, adapted to sunnier, drier conditions. Their fragrances overlap in a few molecules, while separate structures and chemical mixtures give each plant its own scent. The current lilac flowers will soon lose their strongest color. Petals will soften, curl, and fall from the panicles. Leaves will remain on the branches, continuing to gather sunlight and produce sugars through the warmer months. Some energy will maintain roots and add new wood. Another portion will be stored or directed into young growing points. By midsummer, the earliest stages of future flowers may already be forming inside new buds. Their panicles will remain compressed through autumn. Scales will close around them before the leaves fall. Winter cold will hold them through dormancy until spring warmth begins accumulating once more. The flowers that seem to arrive so suddenly are followed almost immediately by the work of preparing flowers that no one will see until another winter has passed.